Hi, my name is Robert Byrne from the Deutsches Herzzentrum München in Munich in Germany, and I'm here at EuroPCR 2015. I'm delighted to be joined by two colleagues of mine, Michael Joner, who's working at CV Path in the United States, and Giulio Stefanini from Milan. Gentlemen, welcome.
Thank you.
Hi, Rob.
So we had the pleasure to be involved in a session on all you need to know about metallic DES during EuroPCR 2015. And that brought up some issues regarding the testing of new metallic DES and bringing new devices to the market. Michael, you have really extensive experience in non-clinical testing of metallic DES. Maybe you could tell us what are the key components that you think we need to be concerned about when we look at new devices?
Yes, so there are some very important components which we need to consider, which is, first of all, non-clinical testing, which is a number of tests, bench-side tests mostly, that will be performed by different institutions. And then we have also preclinical safety tests, which is more where I'm coming from. And in this regard, we have animal studies, which we can talk about. And then there's also toxicology, which is a very important part of these requirements for regulatory affairs.
And what are the key elements of the stent platform that we need to consider when we're doing this non-clinical testing?
So usually a metallic stent is composed of the backbone itself, which has a difference in geometry and stent design, which we need to consider when we plan those tests. Then most often we have a polymeric matrix, which is used for releasing antiproliferative drugs. And we have the drug itself. And so these are the key components that we need to consider. Sometimes it's even more, but in most metallic drug-eluting stents, that's what we have to consider.
Okay. So you're telling us about non-clinical testing in terms of bench work, and then about preclinical testing in an animal model. Metallic DES, what type of preclinical model is most suitable for assessment of new DES technology? And more importantly, what are we looking for?
Yeah, so most of the tests have been performed in the past in the porcine model, simply because we are trying to investigate these devices in an anatomy that is very similar to our patients. So we implant these devices into porcine coronary arteries. What we're looking for essentially are all safety parameters. So we're looking for biocompatibility, tissue response to the stent at short term and also at long term. And finally, we also look at other safety parameters such as re-endothelialization, which we have learned is very important because it relates to the risk of stent thrombosis in our patients.
So you're saying safety is the key issue in these preclinical tests, and we can't learn so much about efficacy or effectiveness?
Yes, that's correct. There is limited information available with regards to the efficacy that we will see later in human trials, simply because we are implanting these stents into healthy arteries which are non-diseased. So we cannot completely predict, but we have some surrogate parameters that will tell us there is a greater or less likelihood that these devices will be effective. And also we will compare them in these studies usually against approved devices from which we have some clinical experience already.
Okay, so when we move on then to address efficacy, I suppose we're moving on to clinical trials. And Giulio, maybe you could tell us a little bit about the assessment of metallic DES in the clinical setting. What types of studies need to be performed with a new DES technology?
Well, Rob, it's actually quite easy. I mean, testing drug-eluting stents in clinical practice means using them and evaluating the safety and efficacy outcomes. So safety means ischemic outcomes such as mortality, such as myocardial infarction, such as stent thrombosis. And effectiveness means instead evaluating the capacity of these devices to prevent restenosis and therefore measuring the target lesion and target vessel revascularization that occur after the implantation of these devices. So where are we heading in terms of evaluation? Of course, when we talk about metallic drug-eluting stents, we're talking about a very mature technology that we are using since several years. Therefore, the evaluation should really start with a simple evaluation and in a relatively small sample size that allows us then to compare the event rates against some standards that come from our previous experience. We call these standards optimal performance criteria, OPC, and mainly comparing the event rates against these standards can reassure us and can allow us to proceed towards a provisional approval of the device.
Okay, so when we talk about clinical trials, I mean, are you saying it's a two-step process that we have at the moment in terms of clinical data?
So, well, at the moment, actually, for approval in the European market, we need mainly to show safety of the device and performance, meaning that the device does what it's claimed to do.
So what size of clinical trials are we looking at in order to get this information?
At the moment, relatively few patients, 40, 50 patients in first-in-man studies. So it would be certainly, what is interesting is that at the moment, who is allowed to certify a device in terms of CE mark is some offices called notified bodies, which are present in almost every European country, and mainly they are allowed to approve device for the EU Commission. Yeah. And what is interesting is that at the moment there is quite some heterogeneity in the evaluation of devices for approval. It would be certainly interesting to see whether it's possible to go in a direction of a higher degree of homogeneity, so that more or less devices are evaluated in the same way in all notified bodies.
So Michael, I suppose you're in an interesting position because you do a lot of preclinical research and you're also an interventional cardiologist. If we look at clinical endpoints, we have heard from Giulio the importance of hard clinical endpoints, but what about surrogate endpoints? How can imaging studies move us forward when we assess metallic DES?
Yeah, I think that's a very important point in the future, simply because our tendency is also to move down to smaller sized studies, and surrogate endpoints in this regard may play a very important role, especially because they provide some translational aspects. For instance, intravascular imaging is something that can be applied in preclinical studies and then also taken to the clinical arena, where we can look for the same kind of surrogates and compare how is the device performing in preclinical studies versus in human trials.
Okay, so what are you talking about? Are you talking about angiography? Are you talking about intravascular imaging?
Both. I'm talking about angiography, but I'm also referring especially to intravascular imaging such as ultrasound or optical coherence tomography based imaging, which is current state of the art.
So I mean, as we move forward, I think we will have clinical trials looking at surrogate endpoints and then trials powered for hard clinical events. Giulio, you're suggesting to us maybe because this is a very mature technology, we can use objective performance criteria, so that would mean single arm studies. Do we need to then move beyond this and perform these all-comer randomized control trials that we're being used to seeing in recent years?
Well, we could imagine a future in which we, first of all, evaluate these devices in small studies, use objective performance criteria, and then provide approval, and which may be a provisional approval, and with the commitment of the device manufacturer to perform a larger trial and compare the device against standard of care, and to get a definite CE approval.
Okay. Well, I know both of you have been involved with myself in an initiative from the ESC and EuroPCR, a task force to look at recommendations for the evaluation of coronary stents, and I know we will have a summary of these proceedings appearing in the near future. So I think we look forward to reading more about these in the near future.
Absolutely.
With that, I think I'll thank you both for being here today. I'll thank you for tuning in to watch us and let you know that there's much more from this session online, the All You Need to Know About Metallic DES at PCR Online. Thank you.